Bismillahirrahmanirrahim. I'm going to review this process of DNA being converted then or copied into RNA, which is then used as a template to make proteins. Because if you review this phenomenon, you actually get an opportunity of reviewing a great deal of the material that we have covered. Now, DNA is, that's deoxyribonucleic acid. Deoxyribonucleic acid. Now, the difference between DNA and RNA, at least even in its name, is the DNA is deoxyribonucleic acid and RNA is simply ribonucleic acid. In other words, this part of this name, the deoxy part, is missing in RNA. So DNA is deoxyribonucleic acid and RNA is ribonucleic acid. And that little nuanced knowledge is actually, if you appreciate it, then, it's kind of cool. Now, if you understand the building blocks of both DNA and RNA are the nucleotides. So, and that you must appreciate. Nucleotide are the building blocks of DNA and RNA, and the constituents of nucleotides are three. They have a phosphate group over here, and if you ask, what is a phosphate group? Well, it's a phosphate group is one phosphorus atom, married to or attached to, 4 oxygen atoms. So, it's perfectly halal. Phosphate married to 4 oxygen. So that's a phosphate group. It's kind of cumbersome to write all those oxygen and phosphate. You just simply write that as a P, as a phosphate group. So a nucleotide is therefore a sugar. Now sugar, this is a pentose sugar, right? So that's a pentose sugar. That means 5-membered ring sugar. And it has a phosphate group in that end, and on this side is attached a nitrogenous base. Now, nitrogenous base is called a nitrogenous base because it has basically boatloads of nitrogen as a part of its constituents, like this. I'm just making this up, and whatever that is, like this. So that's kind of a nitrogenous base. So nitrogenous base, okay, NITR, nitrogenous base. So the constituents of a nucleotide, or what's made of the building block of nucleotide, is the phosphate group, a pentose sugar, and a nitrogenous base. Now, if you recall the properties of sugar, it's like this, and has a lot of boatloads of hydroxyl group like this, and anywhere you don't see something, you put a hydroxyl group in it. The difference between DNA and RNA is this hydroxyl group over there. See, in DNA, there's this hydroxyl group is missing. That's why it's called deoxyribonucleic acid. So the ribonucleic acid, so this is that ribo part of it comes from here as a ribulose sugar. So ribulose is a 5-membered ring sugar, or 5-carbon sugar. And so, and deoxyribulose means it's a ribulose molecule that's missing one hydroxyl group, and that's the name. So deoxyribonucleic acid is one is nucleotides that are missing that one hydroxyl group over there. RNA, on the other hand, has ribonucleic acid. In other words, the sugar that's in RNA will be like this. It'll have a hydroxyl group over here, and it will have a hydroxyl group over here, OH, of course, being the hydroxyl group. So that's the difference between the DNA RNA molecule in terms of nucleotide. Now, while we are at it, let's talk about the differences between DNA versus RNA. Well, that's first, of course, DNA is a double helical structure. In other words, it's like two threads intertwined like that, where RNA is a single-stranded molecule. So one critical difference is that it is a double-stranded molecule, DNA is, and RNA is a single-stranded. The other thing is the 4 nucleotides that constitute all of the DNA, and that is the A, T, G, and C, adenine, thymine, guanine, and cytosine. And in RNA, it's A, U, and GC. In other words, there's no thymine in RNA. So ATGC in DNA, and adenine, uracil, guanine, and cytosine. Wherever there's a T in DNA, there would be a U in the RNA. It would be replaced with a U in the RNA. Now, what's the big difference between the thymine and uracil? This one and this one, not a whole lot. They're almost identical molecules, okay? They're almost identical molecules, but you never find a thymine in RNA. Okay, now, so that's the difference between DNA and RNA. Now, the other aspect of this is that the DNA does not leave the nucleus. So anytime it's kind of like sacred book, like it's a library reference book. You can't take it out. It's just DNA stays in the nucleus. So all the laws that are written in the DNA, it is the genetic code, whether it be the color of the eye of a person, or the skin color, or the texture of their hair, or what molecules they make in their body, is determined by the DNA. So it's kind of like the textbook of the organism, but the textbook is sacred. It stays in the nucleus. Okay, so so if that's the cell, the DNA is going to be restricted to the nucleus. And if you want to use the information DNA, first it's copied and then changed into RNA, and then RNA is then basically takes the message that's in the DNA and gives it and uses it for the purpose that it is intended for. Now, this process, making a copy of the DNA into an RNA, this is called, of course, transcription. This is called transcription. This process is called transcription. And the process of taking the RNA message and making using that as a template, using that as the rules for making a particular protein, this process is called translation. Okay, so the process of making RNA from DNA, that's called transcription, and the process of making proteins out of RNA, this process is called translation. Okay, now this is important to appreciate. Let's see if somebody takes a dictation. So the teacher is giving a lecture and the student is writing the notes. So I say blah 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 blah, and then the student writes blah 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 blah, and then so when the student writes, what the student will write in the same language. So if I write a letter in English, then the student will write the letter in English. That's called transcription. So when somebody's transcribing, then they're just using the same language. They're just writing verbatim what the teacher said. Translation would be taking English and then changing the message to, for example, to Arabic, right? So this is translation. This is an entirely different language, and that makes sense here because DNA is made of nucleotides, right? DNA is made of nucleotides. RNA is made of nucleotides with the slight differences, right? Because of the uracil stuff and the single strand, right? And there's this hydroxyl group missing DNAs. So this is, regardless, DNA is made of nucleotides, RNA is made of nucleotides, but proteins are not made of nucleotides. Proteins are made of amino acids. It's like a different language, correct? So that's why it's called translation. So the process of making proteins out of RNA, that's called translation. The process of making RNA from DNA, that's called transcription. Now, you must understand the basic structure of a nucleotide. Now, nucleotides are the building blocks of DNA, and they're like this, right? They have the nitrogenous base over here. I'm the kind of base that I'm just kind of making it up over here like this type, and then they have the sugar over here, ribose sugar, and the phosphate group here. So that's a nucleotide, right? In the same way, you kind of have to get a feel for the nucleotide. You have to understand what an amino acid is, because you can't mix up an amino acid with a nucleotide. You can't mix up a nucleotide with an amino acid, right? So the basic structure of an amino acid, all amino acids must have this. They must have a nitrogen and a carbon like this. So in this order. So nitrogen, carbon, carbon. No, any molecule that does not have this can't be amino acid. So if it's like this, that's not an amino acid for sure, right? And etc. Or if it's like C and C like this in that order, carbon, nitrogen, carbon, that's not amino acid. It has to be like this. Now the amino acid is called an amino acid is because on this side it has an amine group. Say nitrogen attached to 2 hydrogen over here like this. That's given a name. That's called an amine group. Okay, and as in ammonia, see NH3. This is ammonia. You understand? So ammonia. So this is amine group. Okay, so amino acid has an amine group, and then on the other side it has an acid group. Okay, so here is an acid group like this. So this is a carboxylic acid, if you want to know, if you want to sound fancy. Carboxylic acid, or just you can, for our purposes, is the acid group, amino acid. So this is the amine part of the amino acid, okay, amino, and this is the acid part of the amino acid. This is the amino group, and this is the carboxylic acid or acid group, okay, and that's why it's called an amino acid. Okay, now this carbon is, if that's called the alpha carbon, if you want to know, and whatever's attached to this carbon is what distinguishes one amino acid from another amino acid. If it's like this, it's like glycine, for example. Okay, if there's a hydrogen over there, it's glycine, the simplest of them. And if it's like this, CH3, like CH3, like this is valine, right? So this group, whatever's attached to this carbon here, could be something very simple, or it could be something very complex. This is what distinguishes one amino acid. So we call that the R group. It's the R group, you know, that distinguishes one amino acid. Every carbon has to have 4 bonds. So there's a hydrogen over here. So this carbon has another hydrogen attached over here, which is, you know, to make life simpler. It's understood that it's there, but here, if you wanted to write it, you can write it next to it too. So this is the structure of an amino acid. You kind of have to know it. Like, you know, if you want to draw this, you can say, you know, it's like NH2 like this, and this is carbon like this, and this is like this. And this is the basic structure of an amino acid. And you kind of have to, like, you know, memorize this. You should be able to draw the basic skeletal structure of a nucleotide, and you should be able to draw the basic skeletal structure of a generic amino acid, like you see before you over here. The two are nothing like each other, right? So, and you just kind of have to know, because, you know, you just gotta know. I mean, how can you study stuff and not know this, right? Okay, so that's the amino acids. And so so this process is going from DNA to RNA is called transcription, because they're both nucleotides, and going from RNA to proteins, this process is called translation. And the generic structure of an amino acid that I've described is like this. Okay, so this is a amino acid. Now, as I've suggested already, if this is a cell, and this is the nucleus over here, I'm making it kind of bigger, and the DNA is over here. Okay, so the DNA is kind of like the sacred DNA. It's too cool to leave the nucleus. So if you the data that it carries, DNA is the message, right? It's the code. It's kind of like computer language, 1100100011 type of thing, right? It codes for everything that the cell needs to code for, right? So, but it doesn't leave the nucleus. So you make a copy of it, and then since the two strands of the DNA are complementary, you just make one strand, kind of keeps the message, right? So this is the RNA, okay? And RNA eventually is what's going to leave the nucleus and code for whatever it's going to code for, okay? Now, the couple things I like to suggest to you, when the RNA is first made, like this, Okay, it goes through a bit of a maturation process. Okay, it has some nucleotides that are not necessary for its final functioning. Okay, now why do these nucleotides even exist if they're not necessary, you say? Well, there are several reasons. One reason being is that these might be important for coiling of the DNA in its natural form, and other processes that allow the DNA to do for its proper functioning, replication, etc. So, anyways, there are nucleotide sequences that are not necessary in the RNA. So these are spliced out, okay, and these are taken out. Basically, you take scissors and you splice them out. They're enzymes, of course. They cut those pieces. They're called exons. Okay, and introns. So this part of RNA, this called introns. Exons are the sequences that are excised and removed, and the mature DNA is over here. So the RNA goes through this splicing process, and once it is it's spliced and matures, and it is this RNA that leaves the cell if it has to. Now, I have to say there are different types of RNAs. Okay, that's why some RNA does not leave right away, because some RNA is going to go right over here and make ribosomes. That's which happens in nucleoles. That's what's happening over there, right? So there are different types of RNA. There are 3 types of RNA that we have to get to know. Okay, that's first is the messenger RNA, right? The messenger RNA, and then there is the ribosomal RNA, and finally there is the transfer RNA, tRNA. Okay. Now, the messenger RNA is famous because this messenger RNA is what actually takes the message, the resala of the DNA, the message of the DNA, and that message, which codes for a particular protein, and to be made. See, for example, a cell that produces a pancreas endocrine cell that produces insulin, right? The code for making the insulin is in the DNA. The DNA codes for RNA, and that code, that RNA, is used to string together particular amino acids in a certain sequence to give insulin. So it is the code that dictates which amino acids are going to be sequenced together, attached together to give you that particular protein, for example, like insulin. So of all the various different, like 21, 23 amino acids that exist, right, they have to be stringed together in a particular pattern to give proteins. And each protein is different from each other based on this pattern or sequence of these amino acids. And that sequence of arrangement of amino acids is determined by what code it is in the messenger RNA. And if at the end of this process you appreciate that, if you appreciate how that occurs, you've actually understood some of the beautiful, great secrets of nature that Allah has created. So this is the messenger RNA, is the code that codes for proteins to make the amino acids in particular sequences. The second type of RNA is the ribosomal RNA, and this ribosomal RNA goes on to make proteins. No, I'm just kidding. It goes on to make ribosomes, of course. Okay, so ribosomal RNA is, in other words, if you recall ribosome, if you want to draw a ribosome, it's pretty easy. You draw like this. Okay, ribosome is made of 2 subunits. Okay, it's made of a large subunit, and it's made of a small subunit. So ribosome has a large and a small subunit. All right, and if you were to dive into a ribosome, you'd be surrounded by RNA material, because what the ribosomes are made of RNA. So what kind of RNA? Ribosomal RNA. Okay, so that's the second type of RNA that we shall meet. The third type of RNA is called transfer RNA. Transfer RNA. Now, this is a very important concept to understand. See, now let me just elaborate on this here. See, transfer RNA is funky looking RNA, okay? It's RNA. It's a single-stranded nucleotide material. So, but it looks like this. I'm kind of this is a cartoon picture representation, of course. Hairpin kind of loopy thingy, like this. Okay, like that. So what happens? It's a single strand like this, but it kind of loop a doop and makes like it coils on itself, right? And it's these coils are held together by hydrogen bond between the nucleotides. It's kind of like transfer RNA is like envious of DNA. It's trying to make it like pseudo DNA or something like that, make a double strand like that. But anyways, those are hydrogen bonds that it coils onto itself. What transfer RNA is, in a sense, it's kind of like a taxi, okay, transfer taxi. It's a taxi for amino acids. So right here will be attached an amino acid, okay? So this is a very key concept to understand. See, amino acids are not like floating in the cells. So if you were to dive into a cell, cytoplasm, I know you couldn't really dive into a cell cytoplasm in a thought experiment, right? So anyway, if you were to dive into a cytoplasm, you won't see amino acids like floating in the cytoplasm, right? They would be attached to transfer RNA. So when you eat, food, and you eat the meat, and from the meat you get these amino acids. Those amino acids are digested, and you take them up, and then in the cell they go attached to this transfer RNA. So amino acids are not just hanging out there free in the cytoplasm. They're attached to transfer RNA. Okay. So these are the three types of RNAs that we will meet over here. So in this particular diagram, we'll assume that we're dealing with the messenger RNA. Okay. So once the messenger RNA forms, this messenger RNA will leave the nuclear pore. The nucleus, if you recall, is a double membrane like this, and it will go into the cytoplasm over here like this. Okay. So that's the messenger RNA now in the cytoplasm. Now, as soon as the messenger RNA goes into the cytoplasm, see, the small and large subunit of the ribosome are going to attach. Okay. Now, what's the function of ribosome in the synthesis of DNA? The function of ribosome in the synthesis of DNA, the way simplistically you can understand, is that it's kind of like a workbench. It's where it helps facilitate making of a protein. So as soon as the ribosomes go over here, this is the messenger RNA, right? As soon as it goes, the ribosomes go attached, and that's pretty intuitive to understand because, see, messenger RNA is RNA, ribosome is RNA. They're both nucleotides. If you have two nucleotides together, just like in the DNA, they can attach to each other, right? So ribosomes can kind of like go in there and find appropriate areas where they're attracted to each other, and they just attach over here, okay? Clearly it's more complicated than that, all right? So if you were to really draw a ribosome now, at this stage, we could kind of elaborate on our knowledge of the ribosomes. You could say this is a small subunit and this is a large subunit, right? If you really kind of want to elaborate on that a little bit, you can say, okay, we kind of make this, here's an I, and here's an I, and like this, and like, I'm just kidding that you're going to do that. But these I's that you draw over here are actually important because those are sites of attachment of transfer RNA that we'll see shortly. So those are just kind of like, parking spots, specific sites on the large subunit of RNA. Okay. So this is kind of like ribosome. So once the ribosome attack, as I said, the ribosomes are kind of like the workbench, the desk bench of for making protein synthesis. Now, the protein synthesis is going to occur over here. See, the transfer RNA is going to come and attach over here, and another transfer RNA is going to come and attach over here. Now, if you recall, transfer RNA are fancy taxis for different amino acids. So here's one amino acid, I'm going to make that blue, and here's a different amino acid, I'm going to make that pink, I guess, right? And then these two amino acids are going to bond to each other like that, right? And that's how slowly and slowly you're going to build a protein, and then in this fashion. Now we're kind of getting a little ahead of the story, so kind of recap a little bit, and then we'll go back to the story of protein synthesis. So now, thus far, what we have appreciated is this. So this process from DNA to RNA, that's called transcription. There are different types of RNA. The messenger RNA, which codes for proteins. Ribosomal RNA, which is it goes on to make the ribosomes, the large and small subunit, and that occurs in the nucleolus within the nucleus. And the transfer RNA. Transfer RNA functions as a taxi cab for amino acids. Amino acids are not just swimming in the cytosol pool; they are attached to transfer RNAs. Okay, and then I suggested that as soon as the messenger RNA comes into the cytosol, is the small and large subunit of ribosomes go and attach to it, and they form a platform for protein synthesis. Okay, platform for protein synthesis. Now, to make protein synthesis, right? So what you have to do is you have to take different amino acids there. You can have to string them together like this, right? So these are different amino acids, kind of like, beads in a rosary, like in a tasbih, right? So you kind of have to string different amino acids together. Now, this could be valine, this could be histidine, glycine, valine, alanine, and tryptophan, for example. So you take various different amino acids. If you all string them together like this, after a while, you have a big, long protein like insulin, for example. So, and the sequence of amino acid, which one goes next, is determined by the message that's ultimately in the DNA, which is then photocopied in the form of messenger RNA. So DNA is what ultimately determines the sequence of these amino acids, okay? So to make proteins is to basically attach one amino acid to another acid. Now, how are two amino acids attached to each other? So if you were to take, this is amino acid, if you take this is another amino acid, there's a bond between the two, and this bond is called a peptide bond. Now, this is something the basic. If you don't know that the bond between two amino acids is a peptide bond, you really don't know much about amino acids, period. So you have to know this. So there's a peptide bond here, peptide bond here, peptide bond here, peptide bond here, peptide bond here. So when two amino acids are attached to each other, they form a peptide bond. Now, if you recall, here's an amino acid I just drew for you over here. Okay. And I'm going to just call this one some R1 group for the first amino acid. And then I'm going to draw another amino acid over here. Let me try to do it reasonably neatly over here, okay? There's a hydrogen over here. There's a hydrogen over here. That's a nitrogen, by the way. And there's a carbon, alpha carbon, and that's the carboxylic acid group. And we'll call this one the second amino acid, so R2. R2, okay, now, so those are two amino acids, okay? So when two amino acids combine, the peptide bond that takes place occurs between this carbon and this nitrogen. It's kind of like Lego pieces. See, Lego pieces, there's this one side of Lego piece like this, right? And there's this flat side of the Lego piece. You don't take the flat end and put it against the flat end. You put the flat end to the bumpy end of the other side, right? That's what you do. You take two different looking sides and you attach them together like that. So you take the carboxylic acid group and then attach it to the amino group like this. The next one, you're going to do the same thing. You're going to attach this carboxylic acid group to this nitrogen group. So you're going to take this carboxylic acid group and you're going to attach that. So that's how you're going to string together amino acids. So this is a peptide bond. This is a peptide bond like this. Okay. So now, if you just look at these two amino acids over here, right? All right. So if you just look at those two, I'm going to get rid of the last one over there. So make it a little neater. So the peptide bond forms between here. So the reaction, okay, what you do really is water molecules release. You take that OH group and take that hydrogen group, they leave as water, and that's going cytosol. We are like 70% water anyways. So this nitrogen group, and like here CH, okay, and this is the first R group over there, and that's the carbon over here, and this will be the peptide bond like this and like this between this nitrogen over here, okay, and then there's another, the hydrogen is still, one of the hydrogen is still there, and carbon, and then this carboxylic acid group over here, and that's the R2 over here. R2, okay, so this is the peptide bond right there. See, has that peptide bond. So that's how two amino acids attach. Now this reaction occurs again and again and again, and that's how you string together amino acids to make a protein. And the platform where this happens is this is the ribosome. So our ribosome is basically like a working bench to allow to facilitate these peptide bonds in forming. Okay, so that's what they do. So what happens is now I'm going to go higher magnification of this area right here. So if you have an RNA like this, RNA is made of nucleotide sequences. Let's just say we have, for example, C, A, U, right? So those are it has hundreds of nucleotides, but let's say those are the 3 nucleotides we're focusing on right there. This is the messenger RNA. Okay? Now, a particular transfer RNA is going to come and attach to this over here. It's because it's not just any transfer RNA that's going to come and attach, see? Okay, the transfer RNA that's going to come and attach is going to be complementary to this. So C goes with cytosine, goes with guanine, adenine goes with what? Uracil, and uracil goes with adenine, right? So only the transfer RNA that has complementary nucleotides like this will attach over there. Okay, so this is called the code or the codon. Okay. That's called the codon, and these three nucleotides they are called the anticodon, the code breakers. Okay, so that's the codon and that's the anticodon. Those 3 complementary nucleotides. So every amino acid is attached to a unique transfer RNA. In other words, the taxi for glycine amino acid is different from valine amino acid, is different from histidine amino acid. So they only use their own particular taxi. So every amino acid will only use its own taxis. Okay. And so anytime there's this particular combination of this, you'll find the same amino acid hooked up to that part of the transfer RNA. So this code really determines which tRNA will attach over here, doesn't it? So this is the code which determines which tRNA, because there's a code breaker. Only these particular nucleotide sequences that transfer RNA will stay. So the codon then determines the transfer RNA, which then determines the particular amino acid that's attached to it, right? And the codon was determined by the DNA, see, right? So the DNA determined the codon, which then determined which transfer RNA, and the transfer RNA was attached to a specific amino acid. Therefore, ultimately, this code determined which amino acid was over here. And that's how the specific amino acid sequence is determined, by these 3 codes. Okay, so when the next codon, same thing, the next transfer RNA comes. And when these 2, now think about this, okay? Now you have 2 transfer RNA over here, right? You have 2 transfer RNA with the amino acids right smack right next to. What are they going to do? Of course, they're going to form a peptide bond. That's what they do. So the peptide bond is formed over there. As soon as the peptide bond is formed, this transfer RNA leaves. Okay. And then the RNA, the whole apparatus, ribosomal apparatus, moves over here. And then you will have something like this. Then you have big ribosome unit, small ribosome unit, right? And then you have one of the transfer RNA that's still sticking around over there, like this. Okay, but now it has its amino acid with the peptide bond attached to another amino acid like this. Okay. and then another transfer RNA is going to come and attach over here, and the process is going to begin again and again, and then you're going to string together an entire protein like this. Okay, now this was a bit of a review in sketchy fashion. We should look at this again, this process using PowerPoint slides in some detail. But I kind of want to give you an overview, a review of this important phenomenon, and beautiful phenomenon. I think if you appreciate it. Until next time, assalamu alaikum.